feel somewhat similar to the way it did when we talked about replication, okay? So there's a lot of knowledge that's assumed, some basic knowledge that you guys have from your previous courses. So I'm not gonna hit on every detailed transcription. I'm just hopefully adding to what you already knew and remember from before. So that's what we're looking at tonight. And then on Wednesday, we're gonna start in on talking about, which is chapter 14, I believe, which is thinking about gene regulation. So how do we turn on and off these expression switches? How do we not have our genes just be constantly under expression? And we'll cover that for both, again, prokaryotic and eukaryotic. So those discussions were separating out the main organisms and talking about them differently. Okay. Also, Wednesday night, you will get your exam back. Okay. I know some of you would rather not see it before Thanksgiving. If that's the case, then just don't pick it up. That's fine. My plan is for Wednesday is just to have at the end of the lecture, you guys be able to pick up your exams, take it home, look it over. When we come back from the break, we'll go through all the answers and the detail of that. But for those of you who really want to see how you did, you'll be able to pick that up and get those results on Wednesday. So far, you all know how to map, a circular map. You guys nailed that one. Great job with that. Most people really got that piece. I can see there was a little bit of the struggle in the recap from the old stuff, so remembering that you do have to go back. And that's kind of a good segue for me to, as a reminder, your final is comprehensive, right? So final is a whole entire semester of working material. And we'll chat again when we get to that point, so we'll worry about that when it comes. But so far they look good. The exams don't look terrible by any means. There are parts that make me cringe, but other parts look absolutely fabulous. They look good. So, okay. I think that was it for my announcements. And I'll remind you again on Wednesday, but you do have a homework set that's due also the Monday we get back from break. Okay, so don't go away to break and forget everything. Just eat lots of turkey and lay around on the couch, which is what I would rather do, and I'm sure what you would rather do, but remember you have a little bit of work to do too. All right. Any questions, course stuff? And I also have word, and I'll give you more details as I know them. ASAP tutors are definitely trying to put together a review for the final. I think the only weird piece, and I know there's a ton of ASAP tutors here in the room. You guys don't, it's not open finals week or something like that. But,
the last week, like
yeah.
the very last week of
class instruction.
That's not open.
Right. So I think the review for ASAP, it would have to be before actual finals week for the final for this class.
Yeah, just to elaborate on that, I think the last day of ASAP is the week before finals week, but it's on that Thursday, there's not a Sunday session. So it's the week before on that Thursday, the last day of ASAP.
Oh, wow. So even before like the Friday or Saturday.
Yeah, because we don't offer tutoring on Friday or Saturday anyways.
Or Sundays.
I don't think we're offering it.
Well, I'll update too as we know the details, but there is, it sounds like they want to put together a review session for that for you guys. Okay. Any other? All right, transcription. So we're going to start with bacteria.
I'm just being honest, you don't write that pretty. I like those.
So, as a reminder, when we're thinking about E. coli and prokaryotic systems and transcription, most of their genes are polycistronic. Okay, what that means, that term, polycistronic. Poly meaning many, right? Cistron is actually another term for gene. So when we say prokaryotic genes are polycistronic, what that means is you have one messenger RNA. This is something to keep in mind, that one mRNA will actually produce multiple or many, the poly piece, protein, which which is very unique and different from eukaryotic systems. Eukaryotes, we think about one mRNA is going to be translated into one protein. In E. coli, they transcribe off their DNA and messenger RNA segment that then is translated into multiple proteins. They're all packed together into operons. So we're going to get into a discussion about operons when we start thinking about regulation of these genes. But something to remember, if that wasn't immediately in your knowledge bank. And the other thing is some of these basic kind of segments, if we think about our DNA, our strand of DNA and our strand of RNA, the kind of the main key sites or areas that are on these regions, both the DNA and the RNA, right? Because you end up having a segment that you guys, I know, all remember is called a promoter sequence. So you have some sort of sequence on your DNA that promotes transcription, initiates transcription, makes all the proteins that are going to be involved in transcription start there. So that's called a promoter. And at some point towards the 3' end, you have a terminator sequence. So a way to end this whole process to get RNA polymerase to actually stop. So we're going to talk about what those details are. And then as you start to look at your messenger RNA that may have, again, multiple genes coming off, or sorry, multiple proteins coming off of it, right? We have somewhere along this fragment, you have a site that the ribosome specifically is going to recognize and see. So you have some sort of ribosome binding site. So these are just some general features that we're drawing up here on the board. What else would you have downstream of a ribosomal binding site that we often call +1? What's the generic?
Start codon.
Yeah, start codon, a way to get translation to start. So on our transcript, we have to tell, okay, this is the first amino acid, usually it's ATG, right, methionine. We have some kind of start codon, 3 nucleotides on the messenger RNA that initiates translation there. That's where we start our protein. And by the same token, from our 5' or 3' end, if we're headed towards a 3' end, at some point we have what kind of codon? Stop codon. Somewhere along the way you're telling the ribosome to stop translating. Okay? So a start and a stop codon. So this was all kind of review, easy back into thinking about messenger RNA, DNA, how does transcription work, what are we actually thinking about here, okay? One of the first pieces that I'll expand on from potentially what you had in your prerequisite courses is this promoter region. Okay? So we'll start by talking about specifics of prokaryotic promoter regions. Okay, so for both prokaryotic and eukaryotic systems, we'll talk about how there's a consensus sequence. If I use that term consensus, what does that mean to you guys? What is a consensus? General, but
Yes or a no?
sorry.
Is it like a yes or a no?
No. Yeah, so often we think of, yeah, you're taking a consensus of the class. Do you absolutely hate genetics? Yes or no? And we take a consensus. But that's not, so what does that mean when I'm looking at, if I ask a question of all of you and I pull every single person in this room, gather up that data, yeah, it's more of a majority general opinion. There's data being taken all the time in the United States called it's consensus data. They go around and pull their anonymous pollings that find out how many people live in your household, how much money you make, what kind of job you do, that kind of stuff, and it's pulled into the United States for them to supposedly make good legal decisions and think about how to address the populace based on the consensus. Okay? So for genetics, when we use this term consensus sequence, it means it's like an average, it's like a community poll. They would go through all the genes in the prokaryotic system, the E. coli system, look at their promoter sequence, and say, this is what all those sequences have in common. the main thing to remember for when we get to eukaryotic and prokaryotic, each promoter sequence is different. Okay, for each gene, it's often a different sequence. But there's lots of variations, but we can tend to find this average or these in common parts, this consensus sequence for a promoter. Okay? And it looks from data that's been collected on this, there's two regions that seem to be based on the data, appear to be really important or conserved sequences. So, for example, we would see these two regions common amongst all the promoters, even given the variation of sequences that we see. This is at what is denoted as the minus 10 and the minus 35 position within the promoter sequence. What that means is it's minus 10 bases from the +1 site. So you're moving, I always say backwards, but I know that doesn't necessarily make sense, but you're moving upstream from the translated region, you're moving more towards the 5' in within that, the promoter sequence, so you're moving to the left hand of the +1, those we start to denote as minus negative numbers. Okay. And this minus 10 actually has a special name called the Pribnow box. So again, these sequences can vary from gene to gene, but how closely this minus 10 region, the minus 35 region, so you might see this referred to as Pribnow box or as minus 10. The closer that these match this consensus sequence, this average compiling of sequences, the faster the rate of transcription. So it does make a difference what the sequence is. The reason, or I guess I could put it to you guys, why do you think it would be faster, why would you have a faster rate of transcription if your promoter more closely matches that average sequence?
Recognize faster.
Yeah, the proteins recognize it faster. So RNA polymerase will recognize it much faster. Oh, this is a promoter. I can sit down here, start transcribing. Okay. So depending on the sequence, that can change actually the rate of transcription. And since Mom brought up, she has the guilty look on her face, but since she brought up the concept of we can have a faster rate of transcription because the proteins can find it faster, let's talk about RNA polymerase, which is our main protein involved in this whole transcription sequence, right? So you guys already learned about, thought about DNA polymerases. This is an RNA polymerase. Again, specific to prokaryotic systems, mostly thinking about E. coli. Is RNA polymerase, do you think, one subunit or many? Many subunits, yeah. So this is a huge complex of multi-subunited protein. Okay? There's 4 catalytic subunits on RNA pol, and there's one that's considered a regulatory subunit. So really 5 main subunits on this protein. 2 of them are called alpha. The alpha is, you can think of alpha, I always think of alpha as like being a glue. It's responsible for assembling the core complex. So alpha is more of an assembly type protein subunit. So the entire enzyme is sort of held together and partly assembled, all the pieces, by these 2 alpha subunits. The next type of subunit is a beta. And there are 2, there's a regular beta and a beta prime. So we have 2 alpha subunits, we have beta and beta prime. The betas are what helps this enzyme stick to DNA. So they're a necessary component. If you mutate or take them out of the whole enzyme, this RNA Pol complex will not be able to bind to DNA anymore. So these guys are necessary for binding to DNA. And they also help with the actual RNA synthesis. So making that transcript, which is the main point, right, of having RNA polymerase around. So we have two betas, a beta prime, and then we have two alphas. So we have one last one left. Okay. And this one's the most exciting subunit to me of RNA polymerase. This one's called sigma. And sigma is the actual, sigma is like the main recognition subunit. This is the way that this 10, -10, and -35 are actually recognized by RNA polymerase. So sigma is the one that binds directly to, across that -10, -35 promoter sequences. And the thing about E. coli is it actually has several different types of sigmas. There's many flavors or styles of sigma that recognize different types of promoters. Okay. So E. coli has more than one. And that's how it's able to, that's how E. coli is able to essentially customize its RNA polymerase. If you stick on a different sigma factor, then you can recognize a different promoter sequence and be more efficient at transcribing all different types of genes. So by slapping on a different sigma factor to the same core 2 alphas, 2 betas, now you have a customizable enzyme that can recognize different promoter sequences on different genes. The most common one, the one that we'll spend more time talking about, is sigma 70. So they're often named, some of them have some weird names, but they're often named by the size of the protein. So sigma 70 is 70 kilodaltons in size. This is the major one used in the majority of genes. So most of the genes being transcribed by E. coli at any given time are probably using sigma 70. Okay. So sigma 70 is the one that's actually going to recognize the promoter sequence, and actually shortly after we'll get into elongation here in a minute. So again, reminding yourself that transcription follows similar stages or phases as replication does. You have to initiate process, you elongate the process, and then terminate it, right? So kind of similar to our thinking about replication, transcription and translation follow those same kind of general basic steps. So with initiation, you just have to gather these core subunits together to make the RNA polymerase. Sigma 70 will bind and recognize to that -10, -35 promoter. And then shortly after it starts to move into the elongation phase and create a messenger RNA transcript, sigma 70 will fall off that region. So that's why it can be recycled and used again elsewhere in the genome too. So shortly after RNA synthesis begins, sigma 70 will fall off the DNA. So it's really only neat. Sigma's just there to tell RNA Pol, sit down here, here's a promoter, start transcribing, and then sigma subunit falls off. Any questions about the RNA polymerase subunits that we've covered?
Are these all 5 of them, or are we still going to talk about the regulatory?
No, these are the 5, yeah.
Oh, okay.
Yeah.
Is the sigma 70 the regulatory?
No, sigma 70 is one of the catalytic. So It's more of the beta, one of the betas that's, and I'm blanking right now whether it's beta or beta prime, but I'll look that up for you guys. That's the regulatory. Alphas are catalytic, sigma's catalytic, and then one of the other betas is catalytic, and I'll look that up. Okay. Any other? Okay, so initiation of transcription in prokaryotes, you have your promoter sequence, you get all these subunits of RNA called together, you have them sit down, recognize the promoter region, we start transcription. Transcription starts to go along, elongate, it makes a bubble, just like we think of in replication, where you have to open up those strands, right? So you'll have your double-stranded DNA, and there'll be a small section that's single-stranded, that's our actual transcription bubble. So this is how this whole process of transcription starts to elongate. And the cool thing about RNA polymerase is it can actually wind and unwind the DNA behind it and ahead of it. So it's opening up the DNA but also winding it down behind it. So if this is, if you're following these strands, okay, and this strand right here that we're transcribing is running in this 5' to 3' direction. And we can assign nucleotides to it, just for the sake of practice here. So maybe you have a section on your DNA that's GTAAC. Okay. Your RNA that's coming off of this section and being actively transcribed here is going to be what sequence from the 5' in? So C, A, U, and then a G. Yeah. So it's going to complementary pair as it's being transcribed. It's a complementary base system still. Only every time you would put in a T, normally across from an adenine, you're going to substitute that with uracil, right? So one of the points of this drawing is thinking about the direction that transcription is happening. So it's reading it off. It's synthesizing RNA in a 5' to 3' direction. Okay, it's definitely transcription is moving from the left side of the board to the right side of the board. The RNA pol is reading the DNA in a 3' to 5' direction. So it's reading the template strand opposite the direction that it's headed in and synthesizing it. And it's winding DNA back down behind it. It's crimping this double helix again. It's twisting it up, and it's unwinding the DNA that's ahead of that transcription bubble. RNA pol is doing that. So it's actually retwisting the DNA behind it and untwisting, opening up the way we think of helicase doing for RNA replication. The polymerase itself in transcription is doing this. To 5'. So they're all going to run anti-parallel to each other. Sorry. Here we go. Okay. We're still together here on this. Ish. So what is RNA pol doing? Okay, we know it's has all these subunits. It's adding in new nucleotides. It's basing that off a complementary base pairing to the DNA. Okay? It's specifically catalyzing what bond between what two groups. 5' what and 3' what. 3' hydroxyl. So this is on the previous nucleotide, right? 3' hydroxyl on the previous nucleotide. The new nucleotide it adds in is catalyzing that bond happening on the at the 5' end of it, the phosphate group. Where does it get the energy to do this? How's this reaction fueled? Yeah, melanin. Yeah, any high energy phosphate groups that are being broken and falling off of this, and this really comes from most of the nucleosides. They have 3 phosphate groups on them, and we're going to lose 2 for every catalytic reaction that this happens. So remind yourself again these kind of details that were similar in replication, and we'll see again when you're elongating an RNA versus a DNA. Some of the same kind of chemical details are still going to be the same as in replication, okay? Whether you're building a deoxynucleotide or a ribodeoxy, right?
The beta subunit is the catalytic, and the beta prime is the regulatory. I'll let you know.
Cool, thank you.
So you know.
So beta prime is regulatory, beta is, did I get that right? Beta is catalytic, beta prime is regulatory. Okay. Okay. Does our diagram make sense? We're moving in 5' to 3' direction. It's winding and unwinding the DNA on either side of this bubble. This bubble is not as big as a replication bubble. Okay, transcription bubbles are much smaller in terms of how many actual nucleotide sequences span that whole bubble. Okay. Are there any questions before I move on? So there's 2 main ways that have been studied and researched and described for E. coli to stop this process. Okay, so for termination to happen in prokaryotic organisms, there's kind of two mechanisms I want you guys to know. One is row independent. And sort of by conversely, the other one is going to be row dependent. Okay. We're going to start with row independent. You might see this in your textbook or elsewhere. It's called intrinsic often. Okay. So it's kind of what you should know about this mechanism is it doesn't take any specialized proteins, whereas the row dependent mechanism is going to take a specialized row protein. So row independent is the intrinsic. It's more common. More simplistic mechanism for terminating transcription. Okay. So there are, I erased it, but there are terminator sequences, right, on the DNA. And they vary similar to the way promoter sequences vary, but many of them have around 40 bases of a very GC-rich region, so a region that has lots of guanine and cytosine. And then that 40 or so base pairs of GC, 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 GC is followed by a long stretch of A's. So a bunch of adenines follow this GC-rich area. And what this means, I'll repurpose our little transcription bubble here so we can visualize this. So you have a region on your growing mRNA that ends up having a bunch of these GC bases. And what that means is they can actually hydrogen bond to each other, to themselves. The RNA itself will loop back on itself and make what's called a hairpin loop. And this is made up by a bunch of these bases that are GC, GC, GC repeated over and over, okay? About 40 of them, give or take. And then following that is that whole big run of A's, which means I'm sorry, I wasn't specific about, this is the sequence that's found on the DNA. So if we have a run of A's here on the DNA, that means you have a big run of U's, right? Big stretch of U's on the end of your messenger RNA. So in green here is the mRNA, and blue is our DNA. And this is really I knew when I was first learning this, it was kind of strange to think about, but RNA almost never just sits as a linear fragment nucleic acid. There's almost always secondary structure to it. It folds back on itself. That's really common for almost all types of RNA found in the cell. What happens with this stem loop, though? So this is called a stem loop. The stem loop will actually bind to, it has properties such that it binds to RNA polymerase. Okay? So if RNA Pol, it's kind of hard to depict here, but you can imagine this big enzyme complex. And I'm just drawing it as a dotted black circle. So this big protein sitting in here somewhere wrapped amongst the DNA and transcribing the RNA. So it's a little. The diagrams you see in textbooks and online are always kind of very two-dimensional, simplistic. But we'll repeat that misconception and blob this RNA polymerase enzyme on here. What happens is this stem loop tends to actually bind to RNA Pol. Okay. The stem loop will stick onto and bind to RNA Pol. So once that big GC-rich region is transcribed near the terminator sequence, it forms instantly, curls and forms into that stem loop. The stem loop binds onto RNA Pol and it kind of stops it. It tugs back on RNA Pol. And that stopping, that pausing, okay, normally wouldn't do a whole lot to this whole system, but the fact that it's pausing right when it reached this AAAA-UUUU region, okay, you have a bunch of single hydrogen bonds here. This is a fairly weakly bound region compared to the stem loop, which has all these triple hydrogen bonds. Okay, so you only have the double hydrogen bond there on the AU pairing. So it's a little weaker, and that pausing of RNA Pol will actually pop off the messenger RNA transcript. So once RNA Pol binds to that stem loop, it pauses. And because this is some fairly weak bonding, because you have a huge stretch of A's and U's bound to each other, the pausing actually causes those bonds to be broken. And the whole kind of complex falls apart. So once it pauses there, those hydrogen bonds between the A's and U's, between the messenger RNA, the transcript, and the DNA, they break, RNA Pol will fall off. This is a way to stop RNA transcription, just using the intrinsic properties of the terminator sequence, the fact that it has a bunch of GCs followed by runs of A's. And that's also an inherent property of this polymerase, similar to what we thought of with DNA polymerases. Often when they pause, they fall off the DNA. They need some kind of special, like we remember with DNA replication, we have a clamp that holds on and makes it more processive, makes it stick on the DNA a little bit better. In this case, whenever RNA polymerase pauses, the protein tends to fall off the DNA. Yeah.
Is that because the Sigma 70's gone? Like, is it gone at this point?
And Sigma 70 is gone, totally gone after you pass the promoter region. Yeah, and it's not so. It just is. Yeah, the affinity of the enzyme to the DNA is just not that strong. And transcripts aren't that long. You know, we don't need it to stay on the DNA for really that long, especially in a prokaryotic system where their genes are fairly short.